Now that we know a little bit about meiosis, we can apply that to the process of spermatogenesis. When we look at the process of spermatogenesis, we see that the sperm go through different cellular stages. And the cellular stages of spermatogenesis are seen here. We have Spermatogonia, primary spermatocytes, secondary spermatocytes, Spermatids, and then Spermatozoon, also known as sperm. Let's look at how we apply these cellular stages and meiosis to the process of spermatogenesis. The starting cell of spermatogenesis is the Spermatogonia. The Spermatogonia actually has to undergo mitosis before it can actually go into meiosis. And this is important because Spermatogonia are stem cells. And if the stem cell does not go through mitosis first, we will very quickly run out of stem cells. So the Spermatogonia undergoes mitosis. We get 2 copies of that same cell. One of the copies is retained for more spermatogenesis, whereas the other copy is actually going to go through the process of spermatogenesis. So this newly replicated Spermatogonia is going to differentiate into something called a primary spermatocyte. The primary spermatocyte is truly our starting cell for meiosis 1 in spermatogenesis. But before meiosis can start, this primary spermatocyte has to do something really important. It has to cross that blood-testis barrier. So it actually has to cross past the sustenticular cells of the testes. And it has to get into that protected area within the seminiferous tubules so that it cannot be attacked by the male's immune system. Once inside that protected area, the primary spermatocyte will go through the process of meiosis 1, creating 2 cells that are called secondary spermatocytes. The secondary spermatocytes then undergo meiosis 2, and now they are called spermatids. Spermatids are essentially immature sperm. And these immature sperm undergo a process called spermiogenesis, not to be confused with spermatogenesis. So the spermatids go through spermiogenesis, which is a process of maturation, where basically it becomes a fully mature sperm, also known as a spermatozoon. Here we can see a pictorial representation of the process of spermatogenesis. Here we have our starting spermatogonium, that's singular for Spermatogonia, and it undergoes mitosis, so we get 2 cells. One of the cells is retained so that we don't run out of stem cells for this process, and the other one is going to differentiate, becoming a primary spermatocyte. That primary spermatocyte, which we see here, has to cross that blood-testis barrier in order to have the process of meiosis begin and then complete. Right? So once it crosses that blood-testis barrier, the primary spermatocyte finishes meiosis 1, and then we have 2 cells. Those 2 cells are called secondary spermatocytes. They will undergo the process of meiosis 2, resulting in 4 cells that are the spermatids, or the immature sperm. These immature sperm undergo spermiogenesis and become full-fledged mature sperm.